Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your plant tissue culture medium or media preparation in a plant tissue culture. Media preparation or the component of media for the plant tissue culture. Okay, so first of all, what is this culture media means that? See, culture medium or culture media is simply what? It is the media or a medium for the growth of the plant. Okay, what should we do? We provide a nutrition along with the growth regulator, along with the growth regulator so that the plant will grow and increase their size. Increase their size. So here, what should we are going to do? We are going to discuss about what is the culture medium, what is the component present in a media which is essential for the growth of the plant. Okay. And this plant tissue culture is what? The introductive part of the plant tissue culture are already discussed and it is already uploaded in our account. You can go and check it. Okay. So it is clear what should we are going to discuss. We are going to discuss about the component of the different media which is required for the growth of the plant tissue culture. Clear? Now let's start lecture. Plant tissue culture medium. Okay. Media used in plant tissue culture contain a nutritional component. Okay. Means that we all know that for the growth of everything we require for the growth of everything we required what? We required a nutrition. Okay. It provide the nutrition to the body. That is your nutrition component which provide the nutrition to the body. As we all know that for the growth of human being, this nutrition is important. So for every living being, nutrition is important for their growth and development. For their growth and development. So here, when we required a media for the plant tissue culture, we required what? We required a nutritional component. Okay. Along with the nutritional component, one more factor is very important. That is your one more factor is also important. That is your growth regulator. Okay, it is very important. So along with the nutritional component, we also provide a growth regulator so that the growth or development of the culture tissue take place. Okay, so this totally the tissue culture success is depend upon the type of the media which we used for the plant tissue culture. Okay, if we use a like if we use like high quality of the media, then obviously it will support the growth of the plant tissue culture. And if the quantity of the media or the quality of the media is low, then sometime it will suppress their growth. And suppose that if we have a type of plant tissue culture, and we all know that it will be grow on like suppose that it will grow on a agar media. Now, for their growth, we require this media. If we are not using this media, then the growth of a plant tissue culture will not take place. So, type of the media is important, nature of the media is important, and along with it, growth regulator will also important for the plant tissue culture. Now see what is written in a slide. Media used in plant tissue culture contain a nutritional component. This nutritional component are essential for the growth and development of cultured tissue. The success of the tissue culture depends very much on the type of the culture media used. Okay. Now the next is your each plant tissue culture media contain a following essential to support in vitro plant growth. Okay, so here what we are going to do, we are going to discuss the nutritional component, which are important for the growth of the plant tissue culture, growth and even the development. Okay. So it is important. Now here the list of these factors or list of these component which is essential. Now there are 8 component which are essential for the growth of the this plant tissue culture. And here the plant growth take place in vitro means that inside the lab. Okay. So the component are as follow: macro inorganic nutrients, micro inorganic nutrients, iron as a chelating agent, vitamins, carbon sources, organic nitrogen, plant growth regulators that I already told you along with the nutritional component. This plant growth regulators will also important. And agar. Agar is what? Agar is used for a as a solidifying agent. Okay. So some macronutrients are also important and some micronutrients will also important. Okay. Macronutrients which are required in a larger amount. Okay. And this micronutrients which are required in a smaller amount. So both type of the nutrients are important. So we are going to discuss one by one. Nutrients. Okay, nutrients are divided into two parts. One is your inorganic nutrients and one is your organic nutrients. Okay, inorganic nutrients are again divided into two parts. One is your macronutrients and another one is your micronutrients. Okay, and this organic nutrients are divided into a four category. That is your vitamins, hexitols, amino acids, carbohydrates. And along with this, nitrogen and phosphorus minerals will also important for the growth of the plant tissue culture. Okay, now we are going to discuss one by one of each component. Macronutrients. Okay, we all know that these macronutrients are always required in a larger amount. Okay, suppose that around millimolar millimolar quantity should be present in a plant tissue culture. Millimolar amount should be present in a plant tissue culture. Okay, around 30 ppm. Okay, and these macronutrients are provide both cation and anions for the growth of this plant cells. Okay, now see what is written in the slide. Need of macronutrients is higher. It is present in millimolar quantities more than 30 ppm. Macronutrients provide both anions and cations for the plant cells. Okay, This provide both anions and cations for the plant cells. Okay, now the next is your macronutrients are nitrogen like NO3 and NH4. Okay, now this macronutrients is what? This macronutrients, they are either nitrogen, okay, they are either phosphorus, either potassium, sulfur, magnesium, and calcium. Okay, if it is in the form of nitrogen, then it will be in the form of Na3, NO3, and NH4, ammonium or nitrogen oxide. And if it is present in a phosphorus, then it will be present in a PO4 form, potassium, K form, sulfur, sulfur dioxide, SO4, not sulfur dioxide, sorry, it is SO4. Now, if it is present in the form of magnesium, then it will be Mg. Okay, and if it is present in the form of calcium, then it will be a Ca. Now, macronutrients have a structural and functional role in protein synthesis. Okay, cell wall synthesis, enzyme cofactor, and membrane integrity. Now, these macronutrients have a structural role and functional role, both. They have both type of role, structural and functional, for the synthesis of the protein, for the synthesis of the cell wall, and for the enzyme cofactor and membrane integrity. So, for all these things, these macronutrients are required. Okay. Now, the next one is your nitrogen. These nitrogens are organic compound which are present in the form of either amino acid. Okay, there are different organic acids are present which hydrolyze the Kc. Okay, there are some form of inorganic nitrogens are also present, and they are present or used as a nitrate, NO3, and ammonia, NH4. Nitrogen is also a major or an important component for the plant tissue culture media. Okay, if we take a plant tissue culture media, then for the plant tissue culture media, this nitrogen are very important. Okay, nitrogen are also help in the synthesis of the different organic molecules. Okay, now see what is written in a slide. Inorganic form used as a amino acid, different organic acid, and casein hydrolysate. Inorganic form used as a nitrate or ammonia. Nitrogen is a major component of all plant tissue culture media. Nitrogen are also help to synthesis complex organic molecules. Okay, now the next one. Now potassium. Potassium ions are also present in a higher concentration, and this potassium ion are present in a cytoplasm around 100 to 200 millimoles, and this 100 to 100 to sorry this 100 to 200 millimoles are present in a cytoplasm, and 20 to 200 millimoles are present in a chloroplast. Okay, and it is essential for the ion balancing, activation of enzymes, and also maintain the pressure, osmotic pressure and regulation of osmotic regulation of the cell. Osmotic regulation of the cells. Okay, for ion balancing, for the activation of the enzyme, for the osmotic pressure, maintaining this osmotic pressure, and for the osmotic regulation of the cells. For all these things, we required a potassium ion. This potassium ion are present in a higher concentration in cytoplasm around 100 to 200 millimole and 20 to 200 millimole in chloroplast. Now, potassium ion are essential for maintaining the ion balancing, activation of many enzymes, maintaining osmotic pressure and osmotic regulation of the cells. Now the next is your calcium. This calcium are have a different function and they have a different enzymes which is called a cofactor, and these are bound to the either cell wall or a cell membrane. It also give a strength to the cell wall and maintain the regulation of the cell membrane structure. Okay, if the deficiency of the calcium occur, then due to which there is in disintegration of the membrane are occur and even a shoot tip necrosis is occur. If the calcium deficiency take place, then there are two defect are arises. One is your shoot tip necrosis and another one is your disintegration of the membrane. Disintegration of the membrane. And this calcium is also important for the multiplication of cell and root and supply this calcium are supply as a form of calcium chloride and calcium nitrate. Now see what is written in a slide. Calcium function with different enzyme as a cofactor and it is bound to the cell wall and cell membrane. It give a strength to the cell wall. It help in the regulation of the cell membrane structure. Deficiency cause disintegration of the membrane and shoot tip necrosis. Important in cell and root multiplication. Supplied as a calcium chloride and calcium nitrate. Okay, now the next is your phosphorus. Phosphorus are supplied in the form of phosphorus and it is also important for a energy metabolism. And if the if the deficiency of this phosphorus occur, then it will delay the growth of the plant tissue culture and even a dark green color, um, dark green color of the leaf form due to the deficiency of this phosphorus. Okay, and it will be supplied in the form of sodium hydrogen phosphate and potassium hydrogen phosphate. Okay, and this phosphorus are very essential for the DNA and RNA. Okay, so it is important for the energy metabolism and essential for the DNA and RNA. And along with it, if the deficiency of this phosphorus occur, then it will delay the growth and the leaf of the plant occur dark green color. Now the next is your magnesium. Magnesium are essential for the enzymatic reaction, energy metabolism. That is, energy metabolism means that synthesis of ATP. For these two, uh, this magnesium has two function in the plant tissue culture. One is your enzymatic reaction and another one is your energy metabolism, that is your ATP synthesis. Okay, This magnesium are present in the part of the chlorophyll, and it is supplied as a magnesium sulfate. Okay, it is supplied as a magnesium sulfate and present in a part of the chlorophyll. Now the next is your sulfur. Sulfur are present in the form of the sulfur. Okay, and this is your for sulfur. Okay, and it is also very important substances. If the deficiency of the sulfur occur, then there is an inhibition in the protein synthesis, and it will decreases chlorophyll in a leaf. Okay. If the deficiency of the sulfur occur, then it will inhibit the protein synthesis and also decreases the amount of chlorophyll, decreases the amount of chlorophyll in a leaves. Okay, and this sulfur are supplied in the form of magnesium sulfate and potassium sulfate. It is again supplied in a two form. One is your magnesium sulfate and another one is your potassium sulfate. Now the next is your micronutrients. Now micronutrients, we all know that it is required in a smaller amount, smaller quantities, but if the deficiency of micronutrients are occur, just a minute. Deficiency of micronutrients are occur, then it will lead to disease. Okay. Now, this micronutrients like your boron, manganese, zinc, molybdenum, copper, cobalt, and it is used in a very less amount, that is your 30 ppm or 1 milligram in 1 liter. Okay, and this concentration of this micronutrients are always measured in micromole. Now the next is your zinc. Zinc are important for the protein synthesis. It is important for the protein synthesis and synthesis of the tryptophan. Okay, and then this zinc are supplied as a zinc sulfate. The zinc are supplied as a zinc sulfate. It is important for the protein synthesis and tryptophan synthesis. Now the next is your manganese. This manganese are also play a important role in a Hill reaction of photosynthesis. Okay, now there is a question for you. Write down in a comment box what is the Hill reaction of photosynthesis. Okay, you have a 1 minute for give this answer. What is the Hill reaction of photosynthesis? And for this purpose, this manganese are very important. Okay, it is also required in our enzymatic activities and it will supplied as a manganese sulfate. It will supplied as a manganese sulfate. Now, simply this Hill reaction are defined as a reaction is a light dependent is a light dependent process that occur in photosynthesis When electron from a water are transferred to an electron acceptor. So this is called your Heal reaction of the photosynthesis. Now the next Now these coppers are also play out important role in the photosynthesis. Okay, intermediate of the electron transport chain between our photosystem 1 and 2. If the deficiency of the copper occur, then it will lead to the decrease in photosynthesis and it will supplied in the form of copper sulfate. Now the next is your molybdenum. Molybdenum are essential for the conversion of this nitrate into a ammonium. We all know that in a plant tissue culture or a growth of the plant, this nitrate are converted into a ammonium. Okay, so this molybdenum are essential for this conversion and it is supplied in the form of sodium molybdate. It will supplied in the form of sodium molybdenum. Now the next is your boron. Boron are involved in our enzymatic activities, different enzymatic activities, and it is supplied in the form of boric acid. Now the next is your iron. Iron are important in our enzyme cofactor and it is supplied in a micromole quantities. Okay, and it is used as a supplement with the chelators and complex compound due to its solubility problem. Okay, we all know that this iron have a solubility problem. That's why it is used as a supplemented with the chelators and a complex compound. And it will supplied in the form of Na2FeEDA. Okay, if the deficiency of iron occur, then it will directly affect the growth and development of the plant cell. Okay, if this deficiency occur, then it will affect the growth and development of the plant cell. Now, the next is your organic nutrients. Now, inorganic nutrients are completed. Now, the next is your organic nutrients. In organic nutrient, the first one is your vitamins. These vitamins are essential for the plant synthesis, or it is required for the plant synthesis, and it is also essential for a different biochemical reaction. Okay, we all know that there are a different biochemical reaction take place in a plant tissue culture. So for this reaction, we required a vitamins. Okay, culture cells are capable to produce a vitamin at some level. They required an exogenous supply of different vitamins for a optimum growth. Okay, so culture medium are somehow it is capable that they can synthesize the vitamin itself. Okay, but it can also require a exogenous supply of the different vitamins so that it will take place optimum growth. Now there are some usable vitamins like your thiamine, pyridoxine, nicotinic acid, and vitamin B complex are important for the growth and development of this plant tissue culture. Now the next is your hexitols. Hexitols are important for a tissue culture media and have this compound. It is important for the seed germination, sugar transport, carbohydrate metabolism, membrane structure, and cell wall formation. Okay, and mannitols and sorbitol are a hexitols. Means that these hexitols are supplied in the form of. mannitol, and sorbitol. Okay. Now the next is your amino acid. This in amino acid, glycine are the most common type of amino acid which are required in a different cultured media. And it is essential for the nitrogen containing amino acid which enhance the, sorry, it is not essential, essential, but nitrogen containing amino acid are always enhance the growth and regeneration of the plant. Now the next is your carbohydrate. Cells and tissues are required exogenous supply of the carbohydrate so that it can replace the carbon which the plant normally fixes from the atmosphere by a photosynthesis. Okay, this carbohydrate are supplied in the form of sucrose, and the concentration which is required of carbohydrate for the growth and development of the plant tissue culture is around 20 to 30 gram per liter. Now the next is your gelling agent. Gelling agent is used like your agar and agar. Okay, this agar, we all know that it is what? It is a natural product of the seaweeds, and this since 1658, agar is obtained from a red algae. Red algae from, that is your Geladium gracilaria. Okay, we all know that this agar is melt with water at 100 degrees Celsius and become a solidify at 45 degrees Celsius. And this agar, obviously, it is used as a gelling agent and as a solidifying it. No, in case of this here, we are not using it as a solidifying agent, but as a gelling agent. And this agent properties are totally depend upon the concentration of the agar that we use. Agros. Agros are highly purified agar which are prepared from a Geladium species of seaweed. Agros melt and become a gel at a temperature below 30 degrees Celsius, and it will dissolve during a boiling. Agros are more expensive than a agar. Now the next is your gel right of phyta gel. We all know that just gel right are naturally derived from a polymer, and it is produced by a microbial fermentation of a species, that is your Bacterium Pseudomonas elodia, and it is also a low cost of gelling agent. Around 0.1 to 0.2% are required per liter. Suppose that if we take a 1 liter, then we required it around 0.1 gram to 0.2 gram. Sorry, if we required it 100 ml. Okay, now the next is your natural media. So there are some natural media which is also essential for this plant tissue culture, like your endosperm fruit, coconut milk, fruit material, potato extract, extract of malt, yeast, animal extract, protein hydrolystrate, coconut fruit, orange juice, tomato juice, banana pulp, potato, malt, yeast, fish emulsion, casein hydrolysate, and peptone. They are they are come under the category of this natural media. Plant growth regulator. Okay, after organic, inorganic, nutrient component, we also required a plant regulator for the development of this plant tissue culture. A plant hormone can be defined as a small organic molecule that elicits a physiological responses at very low concentration. These plant growth regulators are also play a very important role in our phenotype and also act as a messenger between the environment and the genome. Now, the auxin are essential for the cell division, cell elongation, and cell differentiation, organogenesis, embryogenesis, callus formation. Auxin is what? Auxin is a growth hormone. Okay, and it is required for the cell division, cell elongation, cell differentiation, organogenesis, embryogenesis, and callus formation. Okay, there are different natural form of auxins are present. That is your IAA, IBA, PAA, and synthesis form of auxins are what NNA and 2,4-D. Now the next is your cytokinin. Cytokinin are also promote the cell division, proliferation, and influence the cell cycle. Embryogenesis inhibit the roof formation. Synthesis in the form of 2-IP, which is more active form of cytokinin. That is your 2-IP. It is what? It is the most active form of cytokinin. Okay. It is synthesized in the form of BAP and natural form of zeatin. Now the next is your gibberellin. Gibberellin promote the stem elongation, bulb conformation, and embryo maturation. And it inhibit the callus for growth and root induction. Uh, it is present in the form of GA3. Abscisic acid, it inhibits the shoot growth and germination of the embryo. And it is thermostable but light sensitive. Okay, means that their properties are not affected when we increases the temperature, but if it has come under the contact of the light, then their property may be affected. So it is thermostable, but along with it, they are what? They are a light sensitive. Now, pH for a tissue culture media, the pH is required between 5 to 5.8. See, it is very important. Otherwise, if you does not maintain the pH, then ultimately the growth will not take place. And your media will not properly prepare. So, before gelling and sterilization. Okay, so we have to maintain the pH before gelling and sterilization with the help of NaOH, sodium hydroxide, QH, potassium hydroxide, HCl, hydrochloric acid. Okay. pH below 5 will not gel properly. Okay, if the pH is below 5, then gel will not form properly. That I have already told you. And if the pH is more than 6, then it will become too hard, means that it will become solidify. Again, gel is not formed, it will become a solidify. So for this purpose, we required a pH maintenance. Morahage and Skog media or MSO or MSO, MS0 is a plant growth medium used in the laboratories for the cultivation of the plant tissue culture. This is the name of the media. MSO was invented by a scientist Tushio Moragic and Paul K. Skong in 1969 during a Morasis search for a new plant growth regulator. Okay, now the next is your here the composition of MS vessel medium. Okay, so here the constituents are present like macronutrients, micronutrient. Here the working concentration and stock regulation. So here the list of this composition of the MSB cell medium. Now the next is your preparation and storage of growth regulator. Okay, so here the data for the how we prepare this growth regulator and how we store it during a tissue culture. Okay, so if we have a auxin, then auxin should be stored at -0 degrees Celsius, -0 degrees Celsius. Okay, and if it is cytokinin, then it will be uh, if it is present in the BAP form, then it will be stored between 2 to 8 degree. If it is present in a Z, then it will be stored at a 0 degrees Celsius. So that's how it is, how we prepare this and how we store it. This is the data about it. Now the next is your preparation and storage of antibiotic stock solution. So here different antibiotic and here it is a stock solution. If we take a amoxicillin, then the stock of it is around 100 milligram per ml in water. and working solution is around 100 microgram per ml, and it will be stored at minus 20 degree Celsius. Okay, streptomycin stock solution 50 milligram per ml in water, and working solution is around 50 microgram per ml, and again it will be stored at minus 20 degree Celsius. Now the next is your media preparation of a plant tissue culture. How we prepare this media. So MS vessel media for a plant tissue culture was prepared in a deionized water, was used, and the various components were added according to the sequence. Okay, now the desired volume of macronutrients, micronutrient, FEDTA, vitamin stock solution were pipetted out in a desired volume in a beaker. Now appropriate amount of plant regulators were added, and desired amount of sucrose or maltose were weighed, added, and deionized water was added to make up the volume of desired final volume. The component were mixed thoroughly on a magnetic stirrer, and then pH should be maintained at 5.8 with the help of either 1 normal of NaOH or 1 normal of HCl. Then semi-solid medium was prepared with the required amount of agar depending on a final concentration. Then media should be autoclaved at 15 psi for 20 minutes at 121 degrees Celsius. Thermolabel plant growth regulator and antibiotic were filtered, sterilized, and then added into a autoclave media when cooled to a below 50 degrees Celsius. And then it is mixed thoroughly in a laminar flow hood. Media were dispensed in an autoclave culture media on sterile petri dishes as per requirement. Now the next, this diagram show how we use magnetic stirrer for mixing the media. So this diagram is represent this is our laboratory. This is the autoclave in which we sterilize the media at 15 psi for 20 minutes at 121 degree Celsius. Okay, this is a autoclave. Now the next is how we pour the media into a petri plate, and this pouring will be done inside the laminar. And here you can see that the proper gloves are wear when we pouring the media into a petri plate. And one more thing, we are not put this petri plate in the table. We have to take it in our hand. So this is a precaution so that it will avoid the contamination. Now this is how we transfer the plant which we want to do a plant tissue culture. So here see that. Okay, now the next here the growth of the plant take place. Now it is ready for transfer into the like outside the environment. So this is all about the media component media required for a plant tissue culture. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for a further detail, you can ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.